chance to host a great scientist and entrepreneur from the UK and the founder of MarraBio, Daniel Peters. He's going to be talking about lots of the cool technologies they've been developing at MarraBio, which was a spin-out from one of the UK universities. So today's webinar is hosted, by the way, is co-hosted by us and TCS Biosciences. As you know, we are supplier of microcarriers, hydrogels, and microfluidic solutions. And TCS is a 60-year-old cell culture supplier in the UK and Europe. And their product really ranges from range of different products, and you can check their website for more details. But they do supply recombinant ACMs that Dan will be talking about today, the cytocaf technologies, bunch of other animal-free recombinant proteins, and as well as our products. Our products particularly are focused on microcarriers, and they range from different options from synthetic all the way to xeno-free options and eatable options for the culture meat sector, along with hydrogels for tissue modeling and 3D bioprinting, and as well as fluidic solutions. Without further ado, I will just pass on the mic to Dan, who's here. We'll go first through a presentation by him, a technical one, and then after that, we can have a question and answer session at the end for about 15 to 20 minutes. Dan, please go ahead and share your screen as well.
Sounds good. Thank you. Right, let me just, there we go. Okay, so yeah, thank you very much for the introduction. Thanks for having me to speak to you all today, and thanks for you all attending. So good morning, good evening, or good afternoon, depending on which time zone you're joining from. So I'm Dan, I'm CEO and co-founder of MarraBio. We're a spin-out company based in the northeast of the UK. And today I'm going to be talking about our cytocaf technology, which is based really on the sort of protein engineering of this quite unusual bacterial protein. And what I'll be describing is how the sort of quite distinct properties of this protein allow us to really develop it for a range of applications and do a variety of different things, particularly with the view of this being a bit like a molecular Lego set, where we can mix and match different sort of flavored bricks to be able to provide different reagents to mammalian cells. So, you know, you may have, probably most of you who are watching this have got some experience of working with mammalian cells, either on small or large scale. And you'll know that really, you know, one of the main goals is to be able to try and recreate or replicate the sort of biological environment that cells would naturally grow in order to get them to sort of grow, proliferate, differentiate, and behave according to the sort of phenotype that you want. And in order to do this, you know, for very simple cell lines, things like hex cells or HeLa cells, you just need to really keep them alive and supply them with the nutrients, get rid of the waste products that they need to keep on living. But for more complex cell types, so for things like, you know, the muscle and fat cells used in cultivated meat, for different stem cell lines, for the immune cells that you need in for example, cell therapies, it gets a bit more complicated because the cells have a greater requirement, particularly in serum-free media, for different biological cues in order to behave and grow correctly. And usually this takes two different forms. For adherent cells, they usually need some sort of substrate to be able to stick down to. And this is usually an extracellular matrix protein or combination of them. For example, MSCs like fibronectin, iPSCs like vitronectin or laminins. And these are very commonly used as sort of coating substrates for either microcarriers or surfaces in order to get these cells to adhere. But they also usually need other factors to, you know, supply them with the right biological cues. And these are usually a cocktail of growth factors. And depending on what the cell needs, these can be more or less complicated. And so they typically contain things like, you know, FGF2, TGF-beta, and possibly some others, depending on what cell type you're trying to grow. And this is all fine, but you know, these reagents really do come with some quite significant challenges. So firstly, a lot of them are incredibly expensive. And this is related really to, you know, the complexity of their manufacture, particularly making them at scale as well. So it's costly to produce, and particularly if you have more than one, and the more complex your cell culture media gets, the greater the expenses in actually supplying these reagents. They also tend to be really highly unstable. For example, FGF2 tends to have a half-life of about 8 hours, meaning it goes off really quickly, loses activity. So usually the way around this is to add more at the beginning, which again increases that expense and complexity. the cells, you need to replenish these growth factors. They're all in the media, so you have to supply them all separately or as a cocktail, and you have to keep replenishing them, which also adds to the sort of labor costs and the complexity of manufacture. And finally, all of this really makes it quite difficult. It's difficult small scale, but trying to get to bigger scale as well, particularly for applications like cultivated meat or for cell therapy, really balloons in both price and complexity of trying to do this. So really, what the goal is that we're trying to solve is to try and see, can we supply reagents to first of all overcome these limitations and supply biological cues in a more sustainable, more sort of scalable, stable format? But also, by doing this, can we also maybe change the way that bioprocessing is done to, again, sort of make cell culture easier and cheaper? And the way we want to approach these challenges is by using this bacterial protein called CAF1, which we've developed then further into our CytoCAF technology. So CAF1 is quite an unusual protein. Its native function is it's a bacterial protein, it's secreted, and it forms a gel-like coat that surrounds the bacteria and protects it from the immune system. In particular, it sort of prevents the bacteria from being engulfed by macrophages of the immune system. And the way it does this is by really being a sort of non-stick, almost Teflon-like coating that basically has a really low affinity for any sort of mammalian cell receptors. And so the cells that are trying to attach to these bacteria, they just can't get a strong hold on the CAF1 coat. And so the bacteria are able to escape and basically not be engulfed by these macrophages. And so it's this non-stick gel-like coat is entirely made of this CAF1 protein. The CAF1 protein itself is actually really small. It's about 15 kilodalton protein. You can see a sort of molecular representation of it there. It's about 2 nanometers wide, 2 nanometers deep, and about 6 nanometers long. So it's small in terms of its weight and also in terms of its physical size. But what this CAF1 protein can do is actually associate with other CAF1 subunits non-covalently to form very long, thin, flexible, and modular polymers. And the way that this works is that the CAF1 subunit, it needs 7 strands to be stable, but it's missing one of these strands. And you can see in that red subunit there, the hole where this strand should be is called the acceptor cleft. And so there's a big gap in the structure of the molecule that needs to be filled. But the CAF1 subunit also itself contains a very sort of extended N-terminal strand, which you can see there in green. And what happens is the N-terminal strand of one subunit fits into the acceptor cleft of another to form a very strong but not covalent interaction. And in this way, these subunits are able to associate into these long polymers. And the polymers themselves can contain 250 or more subunits, meaning that even though each individual subunit is very small, the polymers can be up to micrometers long and megadaltons in size. So they're really quite big. And it has a number of other sort of unusual but very useful properties. So related to its native function, the polymers are inherently biologically inert, meaning cells don't really interact with them in any way. They are able to self-assemble into these polymers. They're incredibly thermostable. So even though they're not chemically linked, they have melting temperatures above 80 degrees, meaning they're incredibly robust. And finally, we make these in just normal lab strain E. coli, but we've been able to optimize the purification and expression procedure really to maximize yields. And so we can make a lot of this, even at fairly sort of low production volumes. And because it's just bacterial fermentation, it means that the cost of producing this is very low. So really, it's kind of an ideal blank slate for protein engineering efforts because it's really stable and there's no inherent bioactivity. And these are properties that would be very difficult to sort of engineer de novo, but as a starting material, very useful for us to be able to modify further. So the core of our technology then really is to search for bioactive factors that people might want to use to grow with their cells, really reduce those to just a very short bioactive peptide motif, and then introduce this at the molecular level into the CAF1 structure in order to make a CAF1-based mimic of this particular factor. So just to take you through that process, then you can see here, this is a structure of FGF2 and as an example of something you might want. And you don't need the full protein to be able to get the bioactivity. You only really need the peptide motif that's able to interact with the cell's receptor and trigger the correct signaling pathways and cell responses. And we identify these peptides at the moment primarily through literature searching, where people have looked at this and published on it before. But there's also a few other technologies that we're beginning to develop to look into this a bit more. And basically, you go through this process, you identify what the bioactive peptide is. You can then introduce this small insertion into the Cap1 structure, but you do it at the genetic level. And that means that when you introduce this plasmid into the bacteria do the hard work in making the protein. You can see that the protein subunit's made there, this time with the addition of the bioactive peptide. And then these subunits self-assemble into these long bioactive polymers. And by doing this, we're able to then create these polymeric mimics. And crucially, the polymers, they can be in suspension, but they also adhere very easily to glass and plastic surfaces as well. So that'll be one of the key themes that comes up when I show you some of the actual data later on, is that these things work in a traditional way, but also in a non-traditional way where they're actually attached to a surface. Now, I mentioned that in the title and in the abstract that we see these as being a bit like a molecular Lego set. And the reason I say this is because the other quite unusual property of CAF1 is this ability to self-assemble following thermal denaturation. So if you take one of the polymers and you do heat it above this 80 degrees, so you have to get it quite hot, it will denature just like most proteins will. And because it's a non-covalent polymer, it will fall apart into its individual subunits. What's unusual, though, is that instead of aggregating like most proteins do, if you cool the solution of the subunits down to room temperature or below, the subunits will refold and self-assemble back into polymers again. And this really gives us quite an interesting ability, which is to take different functionalized modules that contain different bioactivities and actually mix these while they're in the denatured state, so that when the polymers self-assemble, they self-assemble sort of randomly or stochastically to be able to produce a multifunctional polymer that contains a mixture in the ratio that you've put in the subunits of these different bioactivities. So in that way, we can create single materials that are able to possess multiple functionalities at the same time, and then these can adhere to surfaces and remain stable, etc. So that's really an introduction to the kind of the core of a technology. We're able to make, at high scale and low cost, these very, very stable, multifunctional, modular mimics of reagents that are used in mammalian cell culture. So what I'm going to do now is take you through, firstly, just some demonstrations of where we've been able to produce these mimics and kind of where we are with some of these different varieties. And then also some of the other applications that we're looking at in terms of recyclability of polymers, and also how this non-stick nature can also lend itself to some other applications too. So in terms of ECM mimics, and so extracellular matrix proteins mimics, cells like to be able to, adherent cells like to be able to stick down to a surface. And so the choice of ECM protein that you use really depends on the cell type. If you're using mesenchymal stromal cells or MSCs, these really like to stick to fibronectin. If you're using induced pluripotent stem cells, these really prefer either vitronectin or laminin surfaces to be able to stick to. But in both cases, the cells are able to stick down to these surfaces through interactions between their integrin receptors on the cell with their extracellular matrix protein. And the integrin receptors, they have an alpha and a beta subunit. There's a variety of different types, and depending on the type that you have and the dimer that they form, it will basically recognize different motifs. So we can look at the interactions there through these cartoons of these integrin receptors on an MSC with, for example, fibronectin, or on an iPSC with vitronectin. And you can see here that although the receptor structure is quite long and complex, actually the motif they're binding to, the bioactive part of the ECM protein, is quite small. And so really by using the CAF1 technology, what we do is graph these target sites into the CAF1 to be able to make a mimic of these ECM proteins that interact with integrin receptors in the same format, where the integrin receptors are binding to the key motif, but it's within a CAF1 scaffold rather than whatever the original scaffold is, with the CAF1 having all the benefits that I've previously mentioned, including this of just being a lot more structurally simple compared to these other proteins. And so this is kind of what the subunits look like. You can see we've got different positions where we can put the bioactive motif in the CAF1 structure, and also that because the motif itself is quite small, it's not making a major addition to the overall structure of the CAF1. And so the CAF1 is able to retain its properties, the ability to form polymers, the ability to be really stable with this extra bioactivity, but without that addition really disrupting what the protein's doing. So just as a few examples of this then, one of the first ones we made was a mimic of fibronectin, which we've then tested a lot with various cell types, but in particular MSCs. And you can see here, we've compared it to just traditional fibronectin, and it's able to, equally sort of support the growth and proliferation of these cells. The morphology looks normal, and also, you get the expression of the cell surface markers over the different passages that you would expect with MSCs when grown on the fibronectin mimics. So basically, this shows that we can do it, that you can take this motif, graft it onto the CAF1 and produce something that functions like fibronectin and supports the adhesion, growth, and maintenance of the MSCs. Similarly, we've been able to make a vitronectin mimic, which again supports iPSC, adhesion, growth, and maintenance. We did some of this work with the Cell and Gene Therapy Catapult based in London, who saw the same thing, that basically the cells have the right morphology, the right growth, it's maintained over different passages, you have the right cell surface markers you'd expect with pluripotent iPSCs, and also embryo body formation shows that these cells retain the capacity to differentiate into the different sort of germ layer types, which is, again, one of the key features you want when you're growing iPSCs. So in this way, we've been able to produce mimics of key ECM proteins for the growth of these important and quite fussy cell types. And where we are now really is sort of taking this a bit further to look at now more complex bioactivities. So things like growth factors, in particular TGF-beta, we're doing a lot of work on at the moment, and also things like insulin. Again, trying really to target some of the main media components that people use with the goal of then combining these into these sort of single multifunctional polymers later on. And you can see here some of the data that we've got that, the TGF-beta mimic is able to trigger the signaling pathways you'd expect. We see increases in motility, proliferation, all the sort of things you would expect with regular TGF-beta, but in this case, it's in our CAF1 mimic of the growth factor. And similarly with insulin there, you can see that we've been able to use this to trigger the differentiation of MSCs down a more adipogenic sort of cell differentiation pathway with the inclusion of these fat droplets there. And this really, again, shows you that you can take just a small segment of these bioactive factors, put them into CAF1 to be able to make these highly stable mimics. And what I should really highlight here is that actually in both cases, the CAF1 here is actually adhered to a surface of a plate. So rather than being in suspension like traditional growth factors, it's on a surface and presenting this bioactivity in a very different way than usual growth factors do. It's presenting cells with a lot of these signals because it's a polymer, but also, again, it's in an adherent rather than suspension format. And the benefit we feel this will bring is that you don't have to add these things every time you do a media exchange, because these things are attached on the surface, and because they're so stable, it means you don't have to constantly add them, which we think will be really useful for people working with these sorts of factors. So bearing that in mind, then, I thought I'd show something that we've just started to work on quite recently, which is the idea of can we reuse some of these factors? This is mainly, again, with the goal of thinking about things like cultivated meat and other very sort of price-sensitive applications where you're using up a lot of these factors. Everything's at scale. You've got bucket loads of the stuff. And can we reduce the wastage? Again, hoping to reduce some of the costs associated with that by actually reusing and reutilizing some of this material. And the way in which we think that CAF1 is particularly well suited to doing this is in two ways, due to two of its properties. The first one being that because it is so stable, it can survive this process, whether it's room temperature, 37 degrees, whatever. It won't just fall apart while you're working on this. It can last for a long time. And secondly, because of its really long length, because of the large size of these polymers, it means that we can actually sort of retain them by filtration, whereas smaller factors would just pass through. So the idea behind this basically is that you take your CAF1 polymer, you add it to your media, the cells grow, you go through your cell culture process, you then take that media, filter it. Because of the CAF1 size, it's able to be retained, and then you can re-add it to reuse it again in the media, and its high stability ensures that it will still be working. And so we've done some initial proof of concept experiments with this, where basically we took a pot of media, we spiked it with our fibronectin mimic, and then put it through a filtration system and basically see what we got at the end. And you can see here that actually we got really high recovery rates, almost the entire thing we were able to recover with sort of minimal wastage. And you can see from the SGS page here that the protein doesn't really change. It still is polymeric, it's still pure. You don't get loads of other things coming in there, which is brilliant. But of course, the key question is, okay, we can get it back out of the media, but does it still work? And actually, again, the preliminary data from this shows that it does. So again, it's a very simple assay here, just seeing whether you can grow MSCs on the recovered material. But the answer is yes, and you can see there it's got almost the same functionality in terms of the amount of cells that are adhering, and again, the cells look normal there. So this is something we really are keen to sort of follow up on and find out more about, and particularly looking at those more complex bioactivities and doing more in-depth studies as well. But this could be quite big for bioprocessing if it's means these factors can then be reused. We also are looking at microcarriers. Again, this is quite early data here, but what we've done is to coat microcarriers, just plastic microcarriers, with our vitronectin mimic. You can see that we've got some iPSCs growing in and around this. What we've then done is actually harvested these iPSCs, then centrifuged the microcarriers to recover them, and then used them actually and reseeded them with iPSCs. And you can see here that the iPSCs are beginning to grow, and after 5 days of growth, they're still quite happy on these vitronectin coated microcarriers. So in this way, the critical material, which is the coated microcarriers, are able to be recovered and reused, and show this increase, show that they can be reused with these particular cells. So I'm going to just change tack for the last bit of the talk here and talk about some other things that we can do with the CAF1 away from making mimics of other reagents. And that's actually utilizing this other unusual property of the CAF1, which is this sort of non-stick, low adherence nature. And so this is what we've been kind of working on most recently. And we've got two facets to this. The first one is, can we use these low adherence versions of CAF1 to actually make any plastic or glassware a sort of low adherence surface for cells, with a particular goal of actually forming 3D cell structures like organoids and spheroids. So the idea here is, if you've got some adherent cell line, if you just have uncoated plastic, usually, depending on how aggressive they are, they will stick down and proliferate as a monolayer, which isn't great if you're wanting to form a spheroid. Instead, one alternative would be to use a sort of a chemically modified plate, like a low adherence plate, or there's other coatings that you can get. But here, what we've done is actually use this sort of native low adherence CAF1 material. It's a very simple protocol for just coating this, and it sticks down very easily to the plastic surface. And you can see here that now, instead of actually sticking down, the cells stick to each other. Because of this non-stick surface, they can't adhere to the plastic or glass, which favors then cell-cell contacts instead of cell surface contacts, and therefore you get this spheroid nucleation and formation. And it's nice to see a cartoon, but here's the actual data. This is with HCT116, which are a sort of quite aggressive adherent cancer cell line. So we tried to pick quite a difficult target, and you can see here that yes, the cells will stick down on uncoated plastic, but if we coat the cells first with our low adherence cytocaf, the cells don't stick down and instead begin to sort of nucleate and then form these quite nice looking spheroids after a few days. The spheroids themselves seem to be quite healthy. They're not doing anything weird. They don't have big necrotic cores. You can see by live dead staining here. And if we do try and kill them, then they do die, which is good, because again, it shows they're not doing anything weird on these surfaces. And we tried this with a few different cell types. So I've shown you the data with the HCT116 cells. You can see here compared to just using some normal commercial low attachment plates, where we get good viabilities and good looking spheroid formed, in this case with HCT116 and also HEC293 cells. I think we've also done this with HeLa cells and some preliminary experiments with iPSCs too, where we've used it for an embryo body formation assay. The spheroids we see as well, compared to other solutions, we believe that what we're seeing is more actually sort of a greater number of, sort of smaller, more regular spheroids as well. And we're looking at ways of sort of quantifying this a bit better. But in general, the spheroids seem to be a kind of smaller, more circular, more regular sizes. And you can see this back in this picture here, particularly with the HCT cells, where instead of forming these very large kind of contiguous structures where you maybe get one or two very big spheroids, you get sort of smaller, more circular spheroids on our cytocaf coatings. The other thing that we're looking at in terms of low adherence reagents is, rather than stopping them from sticking to a surface, can we also use the cytocaf or the CAF1 to stop the cells from sticking to each other? with really the goal of it being a declumping reagent for cells in solution. And what we can see here with the hex cells is, again, if it's uncoated plastic, the cells will stick to the surface. You saw that if we just have the cytocap on the surface, then the cells don't stick down and form these big spheroids. But if you also add it in the media, you can get these single cell suspensions when used at particular concentration, where the cells quite happily grow in a declumped state. And this is true for hex cells, but also for iPSCs that we've done with our collaborators. And I think one of the key things that we see where we see this being useful is that in traditional declumping reagents, they tend to not be permissive for allowing transfection, which can be a big problem if you want to genetically modify your cells, particularly to produce things like viral vectors. But again, our sort of early stage data shows that the low-adhere anti-clump cytocaf does allow transfection of the cells in the declumped state. And you can see here we've been able to put a GFP gene into some of these HEC293 cells growing in there in the declumps in the presence of the anti-clump reagent. So just to summarize then before we get into the Q&A, what I hope I've shown you then is that we've got a technology really based on the interesting of sort of unusual inherent and engineered properties of this CAF1 protein, where we can create a Lego set of different modules functionalized like ECM proteins, like growth factors, but also in terms of other low adherence reagents, where we can use them for different purposes to try and provide additional or alternate ways of doing mammalian cell culture. They're animal-free, they're ultra-stable, and the protocols are really easy to use. And we can mix and match these different polymers, the different modules to make multifunctional polymers. Our initial data shows that you can recycle and reuse these in cell culture, both from suspension and also in microcarrier formats that could be particularly useful in addressing sort of cost issues in things like cultivated meat. And then also our unmodified reagents are useful as low adherence coatings for particularly 3D cell culture work, but also as anti-clumping reagents for a variety of different cells. And particularly for these low adherence and anti-clumping things, we're just going through the process now where we'd really like to try and get it into other people's hands, see what other cell types these work with. So if you're interested in potentially testing some out, please get in contact with us and we'd be really keen to see what we can do. So thank you very much for listening. I hope this has been of interest and I'll happily take any questions. Thank you.
Thank you, Dan, for the great talk. I think that was really fantastic and really cool to see so many different types of different applications and this Lego sets. I can imagine how powerful they can be. Let's start with the questions in the audience and start from there. So Chloe is asking, how does the CAF1 prevent cells from adhering to each other or the culture surface?
Yeah, it's a really good question. We published a paper on this a few years ago before we span out. So we were looking really at the native function of the CAF1, how it prevents this macrophage adherence. And I think there seem to be sort of, I think, kind of three different ways in which this is able to work. I mean, the short answer is that it seems to be a physical rather than chemical way of stopping the cells from sticking to each other, which is why it's different from kind of other declumping and low adherence reagents. But more specifically, it seems like the first level is there's almost this sort of physical, maybe something to do with the hydration of the protein, where cells just generally don't seem to go near it. So in this published work that we did, we saw that in all different varieties of CAF1, you just got lower cells kind of attaching near the surface or being near the surface in general, regardless of whether they're sticking down or not. The second level is that it seems to have evolved to just not have any affinity or have very, very low affinities for mammalian cell receptors. And the way we demonstrated this was we were able to include one of these integrin binding sites. And when we did that, that kind of allowed some of these macrophages to actually stick to the surface. So in all cases where you used sort of CAF1 versus fibronectin, you got fewer macrophages coming near and being within the sort of range of microscopy in this particular assay we did. But if you used the one which had a high affinity site in, then you did get some cells adhering, whereas in the one without that site, sort of no, or there was very little background. So, and then the third way is that it's incredibly mechanically strong as well as thermally stable, meaning that when cells do exert forces on it, the thing doesn't get ripped apart. So those three methods seem to be the way in which it works. And I think There's probably still some work to be done on working out exactly what that mechanism is, but that's our kind of current understanding of it. Is it's evolved to be low affinity, and also it's got some sort of physical sort of mechanism by which the cells don't really seem to want to come near it.
Fantastic. And I guess another question in that line, Lynn is asking, what happens to essentially to the tagged proteins? Will it change the charges or the hydrophilicity of the protein, or how do they behave?
In general, no. You could see in the picture I put on one of the early slides that actually the addition of these things is pretty small. So in general, they don't really have a major effect, even if they're like really positively charged or hydrophobic or something. It generally doesn't tend to affect the overall properties of the protein. You get some small shifts in PI and things sometimes, but in generally they tend to be the same. There are some that are more extreme modifications that can be a bit more different, but again, the protein in general seems to behave the same way regardless of what you put it in. We've had maybe one of all the things that we've ever tried over the sort of 10 years or so that we've worked on this, both in academia and since we spun out. I think there's been maybe one that has sort of behaved a bit strangely. We're still not quite sure why, but something to do with the chemistry of that tag just interfered with the folding of the protein. But in general, it doesn't really have much of an effect.
That's cool. And I had actually a question there. So what about the stiffness? So if you're coating surface of a tissue culture flask, like without macrocarriers, we've seen a lot of differences on how cells even secrete, not to mention proliferation or elongation with different stiffnesses of substrate. How does it affect the stiffness if you are using this as a single layer coating, let's say with your vitronectin or any other additional like combinations?
That's a really good question. And I think I'm not sure what we did a lot of work before we spun out with hydrogels where we looked at different stiffnesses of that. And I think because it's quite a flexible material, a lot of the differences that we could get with that was by changing things like the cross-linker density and things like that rather than the CAF1 itself. In terms of how what it's like as a single layer, I'm not really sure what the stiffness is that the cells are seeing. We've done some single molecule stuff where we've kind of pulled the subunits apart, looking at the tensile strength of those, but it's not quite the same thing. But what we have seen is that, again, it seems to be very comparable to the proteins it's trying to mimic. So particularly things like the fibronectin and vitronectin, you use them in a similar way and it sort of performs similarly. We don't see any kind of sort of strange effects or anything like that. So I would guess that the stiffness of the substrate is probably similar to other protein coatings. But again, if you want to do that kind of thing, there are ways in which this can be formulated into gels where you can kind of change the overall stiffness of the substrate. But I didn't want to get into that today.
Yeah, no, that's cool. And I guess we had another question on that line as well. Somebody's asking if you have been that this can be an interesting component for bioinks. So I guess you mentioned some hydrogel stuff. Have you ever tried to printing it or applying it to scaffold for bioprinting?
Yeah, absolutely. So one of the really nice things that I didn't go into, which is again related to the long length of these polymers, is that they incorporate really easily into other 3D materials and bioinks as well. So we've done some work which we haven't published, I don't think, but it's based on, for example, like looking at alginates. And the benefit of that is, the alginate, it's liquid, so you can add the CAF and the cells in while everything's still slurry, then add the calcium that cross-links the alginate. And because the CAF1 is so big, it basically gets just physically stuck in there without you needing to kind of chemically modify or sort of, chemically stick it to the rest of the material. So this is great because it means that we can essentially add bioactivity and again, using these multifunctional materials, complex bioactivities very simply into 3D structures as well. We haven't done much. We did some work ages ago showing that it was compatible with printers. We haven't done that much on it ourselves, but some of our collaborators in the chemistry department at Newcastle have got an ongoing project where they're looking at bioinks and stuff. And I think if it's something that is particularly of interest to you, if you if you want to follow up and get in contact, I might be able to put you in touch either with us or with them, and maybe sort of see how things go from there. It's something we're really interested though, looking at how this might work in 3D materials.
Yeah, and I think one other cool thing you shared was the kind of the recycling part of this technology that you can recycle it. How does it work if we were to use that as a detachment strategy? So imagine if we had this coated on a microcarrier, for instance, and we wanted to detach the cells using that recycling mechanism. Do you think that works? Have you tried it before?
We haven't tried it that much yet. I mean, recycling stuff we've only started working on quite recently. One of the things we've also been working on is like looking at making cleavable versions of this, where basically you can use something that's not trypsin or anything like that. You know, an orthogonal enzyme to come in and actually chop basically the key bit that the cell's adhering to off to be able to remove cells in that way. Obviously, that would then destroy the functionality of the protein, but that would be one way of getting it off. We haven't tried using it as detachment yet in seeing, because I know that is a major challenge for people working with these kind of things as well. So yeah, it'd be something interesting to follow up on, I think.
Yeah, no, that's fantastic. And how does the cost compare? I mean, you talked about like culture meat in the past, and wanted to know like, is there recycling bit where you think the majority of the cost saving can come in, or how is it like a head-to-head cost comparison per unit weight?
Yeah, I think it's going to be. I think it's going to be a case of every little helps, particularly with getting towards the kind of price point that cultivated meat needs. I think it's something like $1 or $2 per kilogram of meat is sort of a max that can be spent on these sorts of reagents. So I think to a certain extent we can get it down by at scale we can get it down by a few orders of magnitude just by making things by a simple protein by bacterial fermentation. But I think where we can then go additional, I mean, there's a lot of people working on just making cheaper reagents. Where I think we can go a bit further than that will be in terms of supplying the bioactivity in a different way. So having it adhered on the surface. One of the things we're looking at will be whether you get any increases in potency from that kind of, basically bringing the cells to the growth factor rather than letting it diffuse through, having that polymeric nature of the multiple signals. But also then, as you say, the recycling, where you can maybe get an extra sort of factor of 10 on this because you can use a single reagent multiple times. So I think that we've got quite a lot of scope for really bringing this down by multiple orders of magnitude when people are looking at that kind of scale and those kind of processes. And then looking at some of the techno-economic analyses that are out there to really get further, you get the cost down so much, and then it's all stuff like the structural things that and like investment criteria and other stuff like that a manufacturer would have to work on. So I think that in terms of addressing the actual cost of reagents, we can do quite a lot of that by attacking the problem from multiple angles.
Yeah, that's fantastic. I think that's one of the key factors in biomanufacturing as we are expanding the demand and it's going more and more. We need more and more of the proteins or all these outcomes of these biomanufacturing. That's very cool. So I was very curious also to know a little bit about kind of your journey. I mean, it sounds like a very a roller coaster of different things, like different technologies, different tools being applied together. It's bit of chemistry, a lot of biology, and lots of like background knowledge, I guess, that evolved. So, how was your pathway to your academic slash entrepreneurial journey so far? How did you start, and how did you get here? What did you think of like when first you were learning about Kafwan or you invented it?
Yeah, thank you very much for asking. So it was it came about our CSO Professor Jeremy Lakey, who's a professor of structural biology at Newcastle. He initially started working on the CAF1 as part of a quite a long-term project that was focused on a different area. And as he was doing this, he really began to notice that it had these quite unusual properties. And really, he had the brainwave probably about 10 years ago now of thinking, well, hang on, maybe this could be useful as a biomaterial. And so from that, he was able to get quite a large grant. And the interesting thing about this grant was it was very interdisciplinary. So that was when I joined the project as a postdoc, as well as some of my other colleagues working in chemistry and bioprocessing, and also in cell biology. And so really the idea was to bring all this together and just sort of explore while we're in academia, different things that this could do. And of course, there's a few things that we tried that did or didn't work, but it really kind of allowed us to build up our knowledge base of what this protein could do. So things like the recombination, the making the mosaics, sort of heating, and cooling sort of stuff, all came about as part of that project. But one of the conditions of that funding was always to be thinking quite commercially. I think Jeremy had some previous companies before. When I joined the lab, I was of kind of a pure academic mindset, and really, as the project progressed, I began to think, actually, no, but looking at this commercially, it's really interesting, not just from a kind of intellectual perspective, but also from a career perspective as well. So, as the project progressed, I tried to do some more training activities, particularly one called the iCure program we have here in the UK. which is basically you go out and talk to loads of people, 100 people minimum in different industry, different application areas, really try and work out what are the needs in the market right now. And what was really interesting about that was some of the stuff that I thought this would be really useful for, either wasn't that big a problem, or maybe there was a regulatory barrier, or maybe something else. Whereas actually identified opportunities like large-scale cell culture, cultivated meat, cell therapy manufacturing, things like this, where we could identify problems where we thought, oh, actually, we think our technology could help solve that problem. And so really, it was kind of a route through to that. And we began to realize as a result of that process that we had a platform technology that could do lots of different things, and that really forming a spin out would be the best way of exploring that. And I think, for me personally, it's been really eye-opening to sort of going through that process and seeing actually what was going on in industry, seeing all the really exciting work that's going on in the commercial sense. And I think being able to work on something that you find scientifically interesting but feel could actually be really useful, it's a very different set of challenges, but a really enjoyable one to try and solve.
And what was the point where you thought, okay, I want to definitely commercialize this? Because I guess there will be a lot of people, either postdoc or PhDs, or early career researchers in general, who are inventing new things. But again, there is a point where that kick needs to happen. And it's like you say, like, yep, I'm going to go commercialize this. How was that for you?
Yeah, I remember really clearly. Yeah, I think it was after we'd finished this ICA thing. We'd got the recommendation from a panel of industry experts. This was probably the right course for us to take. I think it was me and Jeremy and our CTO Helen as well. And we were all stood there, and I think Jeremy said to me like, "So we're going to do this then?" And I think Helen and I were like, "Yeah, absolutely." And that was kind of the moment where we decided to go for it. But I think part of that as well was the conditions that helped sort of crystallize that moment, and that was really having all the various support networks and funding mechanisms that were around that to help kind of propel us. So, for example, there was an Innovate UK grant that we were eligible for as part of this ICO process, which would get some money to basically start the business going and do some initial research. There was money to sort of attract other members of the team. We got our executive chairman able to join us through that and help us write the business plan. Various other bits of sort of mentorship network support, training programs that really meant that although it's always a bit daunting starting a business, actually it wasn't, that helped us on that journey because we could see, yes, it's scary, but also there's all these other ways in which we're going to be helped and supported along the way. So let's give it a go and try and get this out into the real world and see whether we can make a difference. So that was kind of a key moment for us.
Yeah, that's very exciting because one thing that I think a lot of the early care researchers will be concerned of is the the essentially you get pulled away from your academic journey. So that obviously that's very competitive itself, especially nowadays with grant writing and applications, scientific publications that you need to have, and other kind of key metrics that you need to hit. So I mean, all these stories so far have been very nice and good to hear, but what were the struggles there as well for you personally? Like, how did it feel? And like, because I'm sure there's a lot of uncertainty there.
Yeah, I mean, I think one of the one of the benefits, thinking about the academic career structure, one of the benefits of being a postdoc is that your time's always limited. You've always got to be thinking about the next thing. So I think there's other people who, for example, were doing some of the same training courses as me, who maybe had to give up permanent lectureship positions or other academic positions. And I think for them, that was possibly a bit of a big question. For me, luckily, my contract was running out anyway, and so this was actually sort of in some ways the next logical step. In terms of the research side of things, I had been thinking about looking at fellowships or other stuff like that. But all of the ideas I had of things I wanted to do next were all really commercial research rather than academic research. And so that kind of helped me to think, actually, yeah, this is probably the right direction to take this. In terms of struggles and the way it kind of changed, I don't really get to spend any time in the lab doing experiments or anything anymore. So I do miss that kind of hands-on aspect. But actually, the flip side of that, which is really nice, is that now I've got this, fantastic team who I work with who are. So it's not just this sort of science that I was producing, which I was interested in, but now it's everyone's science, which is doing stuff that in disciplines that I like, cell biology, which I didn't have any experience in. So actually, it's a lot more exciting, I think, scientifically, in terms of getting to see everyone's stuff and in terms of other challenges, getting those first products out there, learning about how to just set up a company and do things a bit differently. These were all quite big struggles where it was a bit of a, almost culture shock in some ways. But again, they've all been quite enjoyable journeys, particularly the product development where you're getting in the nitty gritty of, okay, well, how does something get bottled? How does it get to a customer? And things like that. And I think, again, the way that we overcame these challenges was really by being supported by people who'd done that before and knew what they were doing. You mentioned earlier, we work very closely with TCS Biosciences. They're experts in sort of liquid handling, fill finish, distributing to customers as well. So being able to kind of have them help us through this and fulfill some of that was incredibly useful. And in addition, we've got a really experienced executive chairman. He's really hands-on, and so knowing how to talk to investors, how to set up systems, how to read the legal documents, all of that kind of stuff. That was how we were able to overcome these challenges. So I guess my advice to people considering a similar journey would be there's an awful lot to learn. It can be quite scary. It can be quite daunting. There's a lot of stuff that you'll probably not have seen before. And the trick is to work with people who are really good, who've done it before, who you get on well with, who can help guide you through that. So I think that's the way we've been able to navigate those challenges.
Yeah, I think that's like definitely a big challenge when it comes to like you're switching your mind from an academic mindset all the way from product development, but setting up a company, trying to have a sales mindset, and then take it step by step. So I totally can resonate there completely. And if you kind of let's say fast forward 10 years, 20 years from now, after MarraBio is successful, do you think you will kind of go back to academia, or you're going to be a serial entrepreneur? What's your long-term goal in there?
It's a good question. I have no idea. I mean, I think it will depend on how things go. I think part of me is thinking that it would be nice to to go back in the lab and sort of get back to kind of discovering and inventing things. I think there's that real excitement of seeing things for the first time and doing that. On the other hand, taking things sort of through and actually getting them into the real world and making that difference, I've found to be I think in many ways more rewarding than just the stuff, the kind of outputs you get in academia. Going through the challenges of grant writing and peer review and publishing stuff, and then you kind of get it out there, and then sometimes it sort of doesn't make as big an impact as you feel that it should do, because there's just so much to read nowadays. I think actually getting something into the real world where you are solving people's challenges and making sure you have that almost that market pull where you've got a real problem you're trying to solve rather than something you just think is a problem. I think that's really attractive too. So it could go either way. I think it'll depend what opportunities we've got at the time.
Yeah, I know. Yeah, that's a very fair enough answer. Now, moving away from kind of starting a company, a lot of early careers, I guess, especially after PhD, they will have a choice between choosing between industry and academia. And we have actually one question in the audience as well. How do you see within UK and maybe Europe, if you're exposed to it, how do you see that pathway different? And what is your advice to those early careers on picking that? Is there a special skill sets that they should find in themselves to go to each of the pathways, or is it more just like pure just passion and so on?
Yeah, that's a really good question. I think there's some key points that I think I'd like to say both from my journey and also from some of the people who have joined our team as well. That coming into this, I sort of did see there being you either stay in academia and you do exciting science and that's great, or you work in industry where you're a sort of small cog in a big machine and it's a bit more secure but maybe a bit less interesting. And that viewpoint was totally wrong. And I think first of all, there's loads of really exciting stuff going on in big industry, which you can get a lot of scientific and intellectual fulfillment from, as well as other things too. But also, I'd missed out completely this sort of SME, small company startup sector, which I think is is really very exciting. And the reason it's nice is because it's actually got, I think, the best bits of both. And this is what I think some of our the first people to join the team found as well, was it was difficult making that binary choice. And that, but actually in this sort of small company, you can do a lot of stuff. You can be really at that cutting edge of doing really exciting science that's really high impact, but not having to just focus on metrics and publications. Maybe having a little bit more stability depending on what the funding situation is like for the company, and also just being a big player in a small team. You've got to be the right kind of, you've got to have the right kind of mindset for it to work for you. But if you are that kind of person, it's different from academia and from particularly different from academia because you're not just working on your own for your own goals. You're part of a big team where everyone's working together towards the same stuff, and so it's really, really collaborative. It's really supportive, at least in our experience of it. And again, you get to do some really, really quite exciting stuff where you're really pushing the envelope. I think where it differs a little bit from academic research, and one of the things to consider is that sometimes it can be um, a little bit more repetitive in some ways, in that you're not making stuff for a publication, but if you are doing that kind of thing, you show something that's in academia, you show something exciting, you show it once, you publish it, and then either you move on or you move forward. I think in the commercial setting, particularly in product development, it's not good enough for it to work once. It has to work every time. And then there's a lot of other stuff you've got to look around that. So, we've done a lot of work looking at the stability of this stuff, which isn't really the most exciting thing in the world, just seeing, yes, is it still there? But the way you have to look at that is actually the outcome of that's really exciting, because yes, although the gel just shows lots of bands that all look the same, if that's the case, then it means that you've got a key feature of your product that you can then take to people and use to help sell it. I think the final thing in terms of mindset of the people, particularly what some of the stuff we look for, is really that kind of enthusiasm and willingness to kind of get stuck in at lots of different levels and help build something from sort of small to big. I think in terms of looking at kind of like technical things, unless there's something really specific that you need, a lot of technical stuff can kind of be trained, or you don't necessarily need someone to come in with all of the experience in the world, but you do need to come in with someone who is just like really open, willing to get involved, willing to work well with other team members, and getting stuck into the other areas as well, because in a small company there's more jobs to do than there are people, and so being able to kind of flex into different things and being enthusiastic about that is great. If you'd rather just do the same thing and just do it really, really well, probably a bigger company is maybe more appropriate, but startups are great for those people who just kind of want to have that bit more get involved in everything kind of view.
Yeah, I think definitely startups is trickier in terms of like you need to actually wear so many different hats on a daily basis. I had a mentor always saying employees within a startup, they should really be like kind of, like there are a lot of jobs that will just get left there that nobody will do. And you need to have that attitude to stand up, pick it up and finish those tasks. And a lot of times about speed, not really quality. Like it actually speed of operation is top priority versus getting one thing done, but very nicely is often not that useful, at least in our experience. So look, that's really great. I think this is very fascinating, and I'm sure we'll be hearing a lot more about MarraBio from the audience and people in the future who are gonna watch this video on YouTube. Please feel free to reach out to Dan. He left his contact details at the beginning of the video, or TCS Biosciences ourselves, and we will be able to connect you if you had any interesting thoughts about their products, you wanted to be a beta tester, or apply their technologies within your research. Anyways, I think this has been a really great talk. Thanks for your time. I know you're very busy, so we really appreciate that. Is there any other thing that I forgot to ask or you wanted to mention?
No, I think that's all good. Thank you very much for the opportunity. Thanks for everyone for listening. And yeah, if you're interested and want to get in touch, please go ahead.
Awesome. Thank you, Dan. And I hope you have a great rest of the week. And I guess it's your early morning. So enjoy the rest of your day. It's evening time for us. So we're going to be heading home soon.
Excellent. Well, have a great evening, everyone. And
Awesome.
yeah, hopefully see you soon.
Thank you. Bye.
Bye.